Hello everybody, this is Dr. Kevin Connors again, and we're going to do a quick video just to help understand oncogenes, tumor oncogenes, and tumor suppressor genes. Uh, it's obviously more information that we can share in a video this size, but just to give you more little hints so we understand what we're talking about. So when we talk about oncogenes, and then there's other genes that are called proto-oncogenes, we're talking about genes that stimulate cancer growth, that increase cancer growth, versus tumor suppressor genes. Tumor suppressor genes do just that. They suppress cancer growth. So as we go through this, I hope you'll understand it a little bit better. So when we look at a normal cell, very confusing picture here, but just look at my cursor here. Look at a normal cell. A normal cell stays a functioning normal cell, and we look at this piece here. This is called a cell cycle. Cells aren't always going through this cell cycle. A normal cell is in a state where it's functioning as a cell. So let's pretend this is a liver cell. So this is functioning as a liver cell, and it's doing its liver cell things. So it's acting out whatever that cell in that tissue is supposed to do. The liver cell, it's a big part of the detoxification piece and helping you detoxify things, etc. Liver does hundreds and hundreds of different functions. That's in a phase called the G0 phase. The G0 phase is how a cell is just functioning normally as its cell. This is the cell cycle, when a cell is undergoing and getting ready to undergo replication. Now, normally a liver cell goes through the cell cycle about, on average, about once every 12 weeks. The purpose is to renew itself and to make another cell so that the mother cell, we could say, then will pass away and go through apoptosis. Your body gets rid of it so that you're always trying to reserve itself, recycle itself, and make a better self to stay functioning as a human being. So normal cells will stay in this G0 phase for a period of time. Some different cells will stay in that G0 phase basically a person's whole life. And then when it goes through this cell cycle of replication, there's different checkpoints along this cycle to prevent excess replication. So in this picture, it's showing excessive growth. This would be a cancer. So this is not supposed to take place. And the way our body has a preventative mechanism to not to have cancer take place is through these different checkpoints through the cell cycle, a replication phase of mitosis. So a normal cell goes through into this G1 phase, and there's certain proteins and enzymes that stimulate this cell cycle phase for G1 to go into the S phase and the G2 phase and the M phase. This is mitosis, where replication takes place. So two cells are produced from this one cell. It turns into two cells, and the mother cell then passes away, goes through apoptosis, leaves a cell left that will stay in the GO phase, hopefully, and until 12 weeks later, it goes into cell replication again. It replicates itself. The mother cell goes through apoptosis. The daughter cell stays in a GO phase for 12 weeks, functions in that cell functioning measure, and then we'll go through this again every 12 weeks. So it's never in a perfect timing like this, but you could say your liver is renewed 4 times a year, maybe. The problem comes in is when a cell is damaged going through this phase, and it is able to bypass the checkpoints, and instead of going into the GO phase and function as a normal cell, it continues through the cell replication cycle and continues to reproduce and reproduce and reproduce. Now, there's things that happen to a cell that cause this. poisons can get inside a cell and damage this mechanism and turn on certain genes that then we call these genes oncogenes. There's several different genes that can be turned on that we will then call oncogenes, the RAS genes, the MYC genes. That these are actually not tumor causing genes per se. These are genes that stimulate growth. So the KRAS system, that would be a RAS gene, is upregulated or increased in case of when I have to heal a wound per se. So when I have to heal a wound, I have to make a lot of new cells in order to heal and scar over that wound. So during a period of trauma, let's say I got in a car accident and I have a bunch of wounds all over me, my normal cell will be kicked in through the KRAS system, through the RAS gene system, that would upregulate mitosis, and my cells will undergo a process of reproduction over and over again in order to heal that wound. And I would be congratulated for being a fast healer, and I'm out of the hospital in no time because my wounds were healed, and this is a wonderful thing. So don't always think that upregulation of the KRAS system is a bad thing. In the case of wound healing, it would be a good thing. In the case of no wound healing is necessary, and a damaged KRAS gene or a defect or mutation on a KRAS gene would upregulate that growth phase, that would be bad in a case of cancer. So now when a cancer gene has upregulated or damaged or mutated KRAS genes, then it's going to stimulate this process and help propagate a cancer. So people with cancer, looking at these different oncogenes, and if the person has mutations or defects on these oncogenes, it would be good to know, because there are some things that we could do to step in to help decrease that. There are some dietary concerns that we could step in to help slow down the KRAS system and the MYC gene system. So these are considered oncogenes in a cancer. They are things that increase the cell cycle replication of cells. Again, everything in the body, we don't want to label it a good thing or a bad thing. It's only good and bad in certain circumstances. So in a cancer, an upregulated KRAS system, someone with a lot of mutations on the KRAS genes, not good to have cancer. So it's going to stimulate cancer growth. The other way that our body protects against cancer growth is what's called a tumor suppressor gene. So there's certain genes that help with these checkpoints. So they look at a cell and see if it's damaged. And if it is damaged, those tumor suppressor genes kick in so that a cell won't go through this process of replication. So if a normal cell gets into the cell cycle and our body recognizes that cell as having damaged genes, then these different checkpoints here should block the replication, and the replication never occurs, and eventually this cell goes through apoptosis. That's our body killing it or recycling it or breaking it down and reutilizing the cell components. So these are called tumor suppressor genes, or our tumor suppressor system. So our tumor suppressor system blocks the cell replication and helps arrest cell function, cell growth function. So P53 is probably, it was the first discovered tumor suppressor gene, and there's much written about that. So that's an example of a tumor suppressor gene. It recognizes a mutation. That P53 gene is turned on. P53 gene releases certain enzymes into the cell, and it can cause an arrest of this replication cycle so that cell doesn't go through replication. It will later help stimulate apoptosis or cell death of that cell, um, or it can actually help repair damaged DNA so that that cell can go back into the GO phase and function as a normally functioning cell. And if it can't, then it will stimulate apoptosis. So the tumor suppressor system is very, very important. So where we see cancer occurring is where the tumor suppressor genes are quote unquote turned off or hypermethylated, and the tumor oncogenes are turned on or acetylated or unmethylated. That's where we have issues, where we have an upregulation of tumor oncogenes and a downregulation of tumor suppressor genes. And that's what's measured in tests like the IVG test. We look at free circulating cell DNA on circulating tumor cells and look at what's the percentage of the oncogenes that are upregulated and the tumor suppressor genes are downregulated, because that would increase them. That would tell us that there's a more aggressive cancer going on. And that's really what gives us the indications. The more aggressive cancer we have, the more oncogenes are turned on and the more tumor suppressor genes are turned off. So we want to make intervention by stimulating tumor suppressor gene upregulation, because we want those suppressing the tumor. And we want to make intervention by downregulating the tumor oncogenes. So hopefully this makes a little bit more sense. We want our tumor suppressor genes working. We want to turn off our tumor oncogenes. There's a bunch of natural things that will stimulate tumor oncogenes. Those are growth factors. And there's a bunch of natural things, stimulators of tumor suppressor genes. So it's important to know those things, and that's what we go through in a lot of our other videos. So hopefully this little diagram gives us a little bit better understanding of tumor oncogenes, tumor suppressor genes, the cell cycle, how that works, how our body has protective measures, and how those protective measures can break down. All right, Dr. Kevin Connors, thank you very much.